All right, so we work at the intersection of cancer biology and immunology, and each of these fields are huge and complex among themselves, and then they have a lot of crosstalk between them. So cancer is obviously super heterogeneous, but at the fundamental level, it's the body cells not behaving properly and also evading the normal regulation of the cells' responses, one of which is the immune system. So normally the immune system is always active, surveying all of the body, looking for things that are going wrong, which may be foreign invaders or things that are going wrong internally. And it's known that the immune system can quickly eradicate very early stages of tumorigenesis. And so the tumors then evolve the ability to avoid or evade the immune system. So this interplay of these two kind of very huge and complex fields is where our research lies. And for those of you who might be interested in further study of these two fields and the history of immunotherapy in cancer, I highly recommend the book The Breakthrough, which is written by Charles Graeber. He's a journalist. He takes a very journalistic perspective. He covers patient experience, as well as the scientists who contributed to the field, both very early understanding of immunology and then up to the more current approvals of immune checkpoint blockade therapy, as well as CAR T therapy. So as I mentioned, the immune system plays a key role in cancer initiation, progression, and survival. And the field of pathology has made clear that tissue biopsy samples hold a wealth of information within them. They're often clinically obtained, they're easy to study, they're relatively easy to study, and so you can get a clinically relevant snapshot of the tumor-immune microenvironment. So the range of what the tumor component is in what the immune component is in any tumor can really vary from very infiltrated tumor. So this is immunohistochemistry staining for the CD8 T cells in brown, and you can see that on this right side, the tumor is hot, so there's lots of immune cells, whereas the one on the far left is cold. There's very little immune content, and what's in the middle is what might be considered warm cells, or warm tumor by some. And so it is logical to believe that this tumor immune component can affect how a patient responds to certain therapies and whether or not can inform the selection of certain therapies. And this was validated with a number of studies now. This is one of the earliest ones. This is actually the first keynote clinical trial in non-small cell lung cancer, which was published in 2015 in the New England Journal of Medicine. This is not work from our group, but just background. But it was found in this case that looking at the marker PD-L1, which is also one of the key targets of immune checkpoint blockade therapy, specifically expressed on tumor cells, did indeed have a correlation with survival. So in the survival curve, the patients who started with a higher amount of PD-L1 in their tumor microenvironment were more likely to survive for longer periods of time. And this raised the inquiry, or the possibility, or the suggestion that these are the patients that you would want to select for immune therapy trials, whereas other patients with other profiling, such as genetic profiling, might be better served with a different therapeutic regimen. So there's lots and lots and lots, but these are just a couple of the key questions in immuno-oncology. So we know that the immune system in the tumor matters, but does it matter in the context of all tumor types? So tumors are obviously super heterogeneous, and it affects all the tissues of the body, and then each of those can have a range of how much immune content there is. Oops, sorry. And then obviously everyone is super interested in the ability to identify immunological regulatory proteins and to determine whether or not they're either clinically relevant as biomarkers or even targetable, so to design rational new therapies in this realm. And so while our assay isn't going to necessarily identify a new target, it's a very good way to test the target, to explore the target, to see how the target is expressed, and to look at the pathways associated with it. And then obviously a big dream would be to be identifying new standards of care, and indeed for several main cancer types, including lung cancer and melanoma, immune checkpoint blockade therapy has become the standard of care over the past 10 years. And continuing to acquire more clinical information and outcome information and tumor immune characterization can help us better characterize, better define improved standard of care. And lastly, and this is the key of what ImmunoProfile was looking for, we want to know if combining genetic profiling with immunoprofiling, will that lead to better patient stratification for treatment, and then also for qualifying for specific trials. So to answer these questions, Dana-Farber put together the Center for Immuno-Oncology. We are led by Steve Hody, who's a melanoma doctor, and ran some of the earliest trials of ipilimumab in melanoma. So we work extremely collaboratively across all of the DFCI disease centers, and we work to correlate, do correlative analysis to identify biomarkers of response, resistance, and toxicity in largely in immunotherapy clinical trials, although we take on a number of additional projects. So within the CIO, we have lots of components. So things start and kind of end with the group that does the consenting and biospecimen collection, and then they are also the people who collect all of the long-term data for the patients. So that's run by Mike Manos here. All of our groups are very integrated with the computational biology group. Obviously, our data set among all of the other data sets are quite large and require pipelines to kind of compress them and make sense of them. So that's led by Jason Weirrather. We have a biostatistics group led by Anita Giobi-Huber, who helps with both the outside of the clinical trial design, how many patients do we need, how will we have discovery cohorts versus validation cohorts, and then also helps with the statistical analysis to correlate any of our data findings with the patient response. The genetics is done with the Translational Immunogenomics Lab, TIGL, which is led by Kathy Wu. And Joanna Baginska runs this immune assessment lab. So I consider them the sister lab because they're usually looking into different compartments, so peripheral blood cells, and looking at the CyTOF, Luminex, Olink, and all kind of immune-related assays, but not necessarily in the tumor and not spatially. And then our group, the Tissue Biomarker Lab, are looking at the spatial aspects of these various tumor samples. So just a little bit more about what we do. So as I've mentioned, we're highly collaborative. We have about 20 consecutively ongoing projects at a time. Right now, we're a group of 8 people total, largely research technicians. 2 of our team members are here today, Nick Weaver, who's a PhD scientist, and Emma, our research operations manager. So our collaborations can have many different flavors. We collaborate, obviously, with DFCI labs and or with specific clinical trials. We collaborate with external collaborators and also industry collaborators, and then we're part of 2 national network collaborations. One is called CIMAC, which I'll touch base on a little later. HTAN, which I won't have much time to go into, stands for the Human Tumor Atlas Network, and that was an effort that was performing both spatial and single-cell sequencing or deeper sequencing assays, kind of modeled after the Cancer Genome Atlas sequencing projects. We look at as being a pan-cancer group, we look at a wide range of cancer types. So currently we have projects in melanoma, lymphoma, glioblastoma, pancreatic cancer, and prostate cancer, and many of our past projects have included head and neck carcinomas, lung cancers, which is both non-small cell and small cell, which are actually quite different disease types within themselves or between themselves, sarcomas, neuroendocrine tumors, and colorectal tumors. On average, we're analyzing about 750 multiplex samples a year, and these data contribute to about 10 to 15 publications each year. So just a diagram of the tumor-immune microenvironment that I thought very nicely highlighted a lot of the cell types that we're looking at, which I have animated with the yellow arrows. So most of our we customize our assays. We can do up to 6 markers at a time, and a lot of people are obviously interested in similar cells. So some of our targets are pretty common. So for example, the FOXP3 marker for T regulatory cells comes up in a lot of our panels. We're often looking at CD8 or another lymphocytic marker such as CD3 or CD4. We're often looking at least one class of macrophages with either CD163 or CD68, and then sometimes including dendritic cells, and looking for how abundant are these cells, how close are they to each other, and then often looking at some of the functional markers to try to establish the status of the cells in the immune microenvironment. And then a lot of our other targets are the key kind of checkpoint inhibitor molecules, PD-1, PD-L1, and CTLA-4, which are all also shown in this diagram. So all of our work, as I mentioned, we work with Scott Rodig. He's trained as a pathologist. So all of our work really kind of starts with core pathology. We look at all of our samples with H&E. They're reviewed by Scott and with us for quality control. And this is first, most importantly, to confirm the tumor type, that what is labeled in the database is actually showing up on the slide. And then Scott will hand annotate the regions of interest that contain the tumor, because obviously any given biopsy can have both normal and diseased tissue. We assess the tumor viability and then the overall suitability of the tissue for downstream spatial analyses, including if there's a lot of fibrotic content and the size of the tissue and kind of the overall integrity of the tissue and the success of the fixation process. We often use single antibody immunohistochemistry for a couple of reasons. One is just overall qualitative assessment. We also use it for validation of all of our immunostaining to ensure IHC is still in the pathology field the gold standard of how protein expression should appear. And then sometimes in the case of the project that I'm showing on the right side, we used MHC1 staining as a way to stratify the patients within a cohort. And so this is a small cell lung cancer project. You can see by H&E already, they look morphologically quite different, even though they're both classified as small cell lung carcinomas. This project was done in collaboration with Dave Barbie's group, and especially Naheed Mahavadan, who is a pathologist himself. And by IHC, so normally MHC1 is expressed on every cell of the body, and tumors typically retain MHC1 expression, but one major mechanism of avoiding the immune system is by downregulation of MHC1. And so that was seen in a subset of the patients in this cohort, and then the MHC1 staining was then used as a mechanism to have two different populations and then evaluate how those patients in each cohort responded to therapy, and those data are published in Cancer Discovery 2 years ago. So the most of our time is spent doing multiplex immunofluorescence. We obviously use the Akoya system because we're here. So so we use opals and the PhenoImager. We can do up to 8 markers at a time. Most often we do 6 markers, and that's what we're comfortable pitching to most collaborators. It's just a little bit more flexible and certainly easier to analyze. So far we've probably optimized over 30 panels. Again, many of those are incorporating the same markers, just changing channels, changing run order, that type of thing. So there's kind of a core group of immune markers that come up a lot, and then we'll customize in any antibody that helps an investigator specifically answer their question. So for quantification, we use the Akoya Inform image analysis software. So this is how my group spends most of every day in the lab. So the steps that are carried out there are that the images are spectrally separated, and then a segmentation map is created there using the segmentation features in Inform. The fluorescence is quantified for each channel. We almost are always using a combination of the 2 main capabilities that are built into Inform. I don't know how many of you use it, but so one of them is called phenotyping, and that's a machine learning feature where you can train the cells that you want to be selected as positive for any feature. And then scoring is setting a threshold based on setting a threshold to define positive and negative cells. So after we do the main Inform analysis, we export those data, and then we send them along to our CIO computational biology group that I mentioned, and is led by Jason Weirrather. And they do what we call data extraction, and this gives us back kind of more manageable set of data rather than single cell level data of populations, percent data, co-expression, et cetera. And then after that, in collaboration with the data group that we're working with, we'll send the data to Anita for additional data analysis and correlation to outcome. So as I mentioned, we're part of two national networks. I just wanted to touch on CIMAC because I think it's a really interesting premise. So this stands for the Cancer Immune Monitoring and Analysis Center. And like in CIO, it's part of both of the other CIO labs of the genetics and the IAL are also involved in CIMAC. And so what CIMAC was designed to do was to set up a set of correlative assays, one of which is our multiplex assay, that are harmonized at the four different institutions, which are Dana-Farber, Mount Sinai, MD Anderson, and Stanford. And so any national immunotherapy trial can apply through the NCI for us to carry out, for any one of the four institutes to carry out any of those four assays. And so far, our group has performed multiplex immunofluorescence for 6 trials, and 2 more are pending. And they've spanned a wide range of cancers, including colorectal cancer, acute myeloid leukemia, breast cancer, and pancreatic cancer. A number of these have already been published. So the first trial that we published was with John Schonfeld, who's at Dana-Farber, on colorectal cancer, came out in 2021. We then followed up a study with William Gillanders in Washington University on triple-negative breast cancer. And then more recently, earlier this year, we published a paper on a very big cohort of acute myeloid leukemia patients that had over 150 samples, partly because there were multiple time points per patient. And that was published, like I said, earlier this year. And so the current CIMAC collaboration that we have going on now, which is actively being stained and analyzed, is called Trial 10208, and it's a pancreatic cancer trial. It's a relatively small patient size, and then it's a complex trial because it has 3 arms of combination therapy and it's dose escalation. It's run by Anna Sperfico, who's at the University Health Network in Toronto, and Princess Margaret Cancer Center. So the design of the trial includes biopsy collection at 2 time points, so cycle 1, day 7, so 7 days after their first dose, and cycle 2, day 7, 7 days after their second dose. And what she's really interested in seeing shifts in the immune populations between time points. And in this case, we have samples that we're getting for the tissue assay, and the IAL group is also getting blood samples for these times, so we'll have a pretty rich data set at the end. And our 6-plex harmonized assay for this trial includes CD3 and CD8, PD1, PD-L1, FOXP3, and cytokeratin. And this is some of the recent staining that came off. So we have very highly inflamed controls. We have a very highly inflamed tonsil control and a known head and neck squamous cell carcinoma that we use as a control that we know has an abundance of tissue. But you can see that it's a lot of wealth of immune cells in this area, and these are the single the single channels of each of the images. And you can see we're getting very clean staining, very little crosstalk between channels, and very robust. So this, as I said, these samples, we're actually still receiving some of the cohorts. The first 2, I think, have been stained, and we're working through the analysis. And as Harini mentioned, we've worked on an assay called ImmunoProfile. So this started before my time in the lab. Scott Rodig and Kristen Felt really spearheaded it, among others, many others. And so it's working toward the idea that a clinical assay will be very advantageous over the standard kind of pathology review of now that is very time-consuming. So it's multiplex immunofluorescence, staining of FFPE tissues. The immune markers that are included in the panel are CD8, PD1, PD-L1, and FOXP3, our favorites. The panel, so then we can use one of 3 different tumor markers. Cytokeratin is the marker that's used for most solid tumors. Pax8 is a smaller class, but it can be used for renal cell carcinomas or thyroid cancers. And then SOX10 is a melanoma marker. So as Serena mentioned, we have over 2,000 samples that have been completed, and they had paired sequencing through Dana-Farber's Onco panel sequencing are available for about half of them. And just some examples of what things look like in ImmunoProfile and how the workflow went. So the data, so there's kind of two versions of the viewers. So I'll be showing you the one that we hope is going to become publicly accessible once it's published, but this is the working one. But you have the whole side scan with the regions of interest indicated here in the red boxes, and then the ROI level image of just the staining, and then how the inform phenotyping looked. So this sample had very low inflammation. And one of the things that we really like about the ImmunoProfile is we can kind of rank where all the samples fall among the full set of 2,000. So this one kind of ranking in the low 20s or mid 20s is obviously not very inflamed. In contrast, this one was actually the most PD-L1 positive tumor that they had in the cohort. It's also highly inflamed with CD8s being in the 95th percentile. And again, the whole side scan and individual ROIs are shown. So as I mentioned, so there's now, the data from ImmunoProfile have all been uploaded and are correlated with the OncoPanel data in a resource that's called CBio Portal, which is currently only available to Dana-Farber, but is meant to be made public when the data are published, and the work is obviously on its way to being published. And as Harry mentioned, we have a number of different cancer types that are represented. These are the numbers of each of the tumor types that are in the data set. And this is just summary statistics of the CD8 and PD-L1, kind of the bins of how the samples appeared. The viewer also includes all of the actual images. So the H&E image with the original pathologist annotation of where to find the tumor, the whole side scan with the ROIs that were selected for analysis, and then each ROI, which can be viewed both as the actual staining, how it appeared, and how the quantification was made. So ImmunoProfile had a lot of interest, but one of the limitations was it was really only accessible to prospectively collected patients. And so a lot of investigators were interested in being able to do that kind of staining on retrospective cohorts. So one example of an investigator that came to us with this interest is Glenn Hanna, who's a head and neck oncologist at Dana-Farber. And he had an instance where he's really interested in oral leukoplasias. So these are diseases of the mouth and kind of throat. And noticed, and it's known that there's different classes. One's called localized leukoplasia, LL, and one's called proliferative leukoplasia, PL. And the outcomes for these patients, these are survival curves and then a cancer-free survival curve, are very different. And so he wanted a way to identify patients sooner rather than later, to stratify them for more aggressive therapy, and also wondered if the immune content of these two different types of dysplasias were different, and if and that might suggest a rational approach or a rational reason to try immune checkpoint therapy on these types of patients. And indeed, we did research. So we carried out a version of ImmunoProfile called Research ImmunoProfile in our group at the Tissue Biomarker Lab. And we found that indeed, the proliferating leukoplasias here in orange were statistically more significant in the PD-1 content and the CD8 PD-1 content. And so this did suggest that targeting the PD-1, PD-L1 axis could be a good candidate for treatment for the proliferating leukoplakia cases. And Glenn Hanna has gone on to start a trial to test nivolumab, which is an anti-PD-1 therapy in this disease. So it's ongoing, still being enrolled. So that's the main data that I had. I hope that I've shown you that multiplex immunofluorescence is a powerful way to quantify the immune cell densities within tumor samples. And we strongly believe in our group that characterizing the tumor-immune microenvironment of cancers enables lots of things, including the more effective use of standard therapies, the more effective development of new therapies, more efficient clinical trials and better trial design, and more meaningful basic cancer research. So thank you all for your attention. Definitely did better than the middle schoolers. And also lots of people to thank. Our current TBL team is really great. Everyone's awesome to work with. Scott, Nick, Emma, Steph, Billy, Naomi, Catherine, and Harry. The ImmunoProfile team, I mentioned Kristen already, but both Bajaya Sharma and Aviza Genji were instrumental in that role, along with many members of the Brigham Women's pathology team, and also huge thanks to the DFCI Knowledge Systems Group, who are the people who transferred the data into the CBio portal for viewing. I mentioned the Center for Immuno-Oncology is run by Steve Hody, and the associate director is Shrin Rana Singh. And then our computational bio team, for whom we would never get interpretable data without our Jason, Ian, and Allison. And happy to take questions, of course.
Any questions?
Yep.
Can you just comment on how long it takes you to do this and kind of workflows?
Yeah, of course. So certainly there's sort of two slow parts, but I still think the slowest part is optimizing a new panel. So for us, that takes several runs. So we do our staining with the Bond RX. So that staining will run for the 6-plex panel overnight, and then the Polaris will stain it the next day. And then, we select the images, we select the ROIs, and then the analysis on the computer, I like to say it takes us 2 hours a case. Sometimes it's more, sometimes it's less. So we have, and then with the group that we have, it means mostly everyone get kind of 1 night run in a week. So then they're getting if it's a full case run, that's 18 cases. And on average, that takes at least 2 weeks to sort of process through. So we kind of have a staggered workflow. And the cohorts that come to us are quite different in size. So a lot of people will come with like 5 really important samples, or 1 sometimes, but a lot of people come with 20 or 30, but then other groups come and they have 150 cases that they want us to look at. And so then that takes, and there's the battle, these assays are very information intensive and slow to carry out. With science always wants more, right? So they're used to being able to get 2,000 patients of sequencing data and then make conclusions from that. So that's a difficult balance.
Have you ever experienced code localization with the immunomarkers? And also, what is your analysis software for doing the actual scoring? Are you doing phenotyping as well?
Yeah, yes, we definitely see co-localization in some, and part of the reason why optimization is one of our slowest steps is that we know that sometimes you can have problems if kind of your first marker is too rich on the cells. Sometimes the second one doesn't stain as well. So part of the reason why I showed the CD8, CD3 panel was we had to re-optimize it because we were starting to see more and more conflict with that. But then we worked really hard to make some adjustments to how we were doing the staining to ensure that we were getting the proper co-localization. And then in our main panel, we always have CD8 and PD1, and PD1 will be on CD8. So we always checked to make sure that the staining is working. So we always use the Inform software, and we do the phenotyping and scoring for almost everything.
Awesome. Thanks, Kathy. I have one question from the chat in Zoom. I'm going to read it out to you. Jian Chen asks, besides utilizing well-validated markers and staining antibodies, have you tried using new markers identified by differential gene expression analysis to stain the tumors? How easy is it to set up assays with new markers?
Yeah, so we definitely have done that some, and I would say on average it's pretty hard, and I think that's because having the assays are dependent on, I think, really good antibodies, and the better your antibody as a reagent, the better the staining is going to be. And so a lot of less defined molecules that are coming out of a gene expression analysis that are newer molecules, it's hard to get that quality of a reagent just kind of right off the bat. But once when we do the validation by doing IHC first in tissues where we expect to see the staining, so we get a sense of the staining, and then we do single IF. For the most part, if we can get it to work in brown staining, we can get it to work in IF. If we can get it to work in IF, we can manage to get it to work somehow into a panel, not always with all the markers that we want. And I feel like I forgot a little piece of the question. So it is something that we do a fair bit. There's challenge to it, and it comes up all the time that people want.
New markers.
Yeah.
And you said that you had already many panels that you have already optimized to go. So given that you tested more than 10 cancer types, are there different panels you use for different cancers? Or is it, you start with the immunoprofile panel and then go about like distinguishing different cancers if required? Or is it just something that you offer based on if somebody asks for a specific panel?
Yeah, it's mostly with the conversations with the collaborator. What are they looking for? What are the goals of their study? Does research immunoprofile make sense as the panel to test? Does a tweak of that panel make sense, or do we need to do something different? And what immunoprofile didn't, it's kind of ironic because Scott is actually a hematopathologist, so he studies blood cancers, but immunoprofile they never got the full validation for the blood cancer tumor markers, and so it's not part of immunoprofile. But certainly we run a fair number. Scott's a hematopathologist, so clearly, I mentioned some leukemia projects, we run a number of lymphoma projects. And so those types of diseases do require kind of more novel panels and more nuanced development of the markers.
Awesome. Any other questions? Okay. Thank you so much, Kathy. This was a great talk.